Sattar, working as assistant professor in JSS College, Dharwad. Today, I like to share my knowledge about sex determination topic, and let us get started with the video. In the previous class, we discussed about the chromosomal mechanism of sex determination. Chromosomal mechanism of sex determination, in that we have discussed in detail about four types. Number one is XX and XY mechanism of sex determination, XX and XO mechanism of sex determination, then ZZ and ZW mechanism of sex determination, and lastly, that is ZZ and ZO mechanism of sex determination in different species animals. In continuation with that slide, today we shall try to focus on the next few concepts in this topic. The next concept is a special case of sex determination in man and Drosophila. Next to that is the Gynandromorphs and Intersexes formation, and lastly, that is the environmental as well as hormonal role in the determination of the sex, and lastly, that is amniocentesis. And now we shall start with the first concept, that is the role of chromosomes in determination of sex in case of Drosophila and man. As we know it already that XX and XY mechanism of sex determination is made possible to determine sex in both Drosophila as well as man. You all know that normal females have XX chromosome complement and normal males have XY chromosome complement. Diploid human cells, they carry 23 pairs of chromosomes or 46 chromosomes, whereas the 22 pairs of chromosomes are known to be autosomes, and the 23rd pair or the last pair is known to be sex chromosomes. If it is XX type is for the femaleness and XY type is for the maleness. And here in man, the presence of Y chromosome makes a difference. Why? Because it carries the set of genes for the differentiation of gonads into testes as a maleness. What kind of genes are located on the Y chromosome? Particularly, major type of gene present on the Y chromosome is known to be SRY gene or TDF gene, that is testis determining factor, known to be a master factor because it triggers the remaining genes to express for the appearance of the male phenotypical characters. So here this is more important. In this way it is possible to determine sex in case of man. Moving on to the next slide, already I said that here is Y chromosome known to carry the master regulator as a TDF gene, and it triggers the expression of large number of genes that produce male sex phenotypes. Moving on to the next one, the determination of sex in case of Drosophila, that is by referring XX and XY type or mechanism. How? As usually you know that in case of Drosophila, females are homogametic, where they carry XX chromosomes, they are enough. Whereas the males have different chromosome complement, as I said before, that one rod like X chromosome and another one hook like Y chromosome. But it is later on known that Y chromosome doesn't play a significant role in determining the sex of the Drosophila as a male. Then how that is made possible? Then what is the role of Y chromosome? Let me make it very clear that Y chromosome, whatever the genes it has, is meant for involving and expressing the fertility capacity of the Drosophila in male, but not for the expression of male sexual characters expression or the development of male Drosophila. Then we need to know the special case by referring a theory proposed by C.B. Bridges, that is known as the Genic balance theory. Genic balance theory tells us that, or it makes us, or it enables us to determine sex, how maleness is expressed in case of Drosophila. According to this genic balance theory, we need to consider the autosomes along with Y chromosome for the expression of maleness. Let me tell you very clearly by showing the next slide. Sex index or sex, that is as a male in a Drosophila, is determined, or it is understood by considering a ratio of number of X chromosomes to the number of set of autosomes. Please try to understand something. It is a tricky or different from the previous type, that is what we have understood in man. If it is XX complement, chromosome complement, obviously that will develop into a female Drosophila. But as we know, Y has no role to express the maleness. Then how that is possible? Whatever the genes to express maleness are, instead of presenting on the Y chromosome, they are present on the set of autosomes present in the sperm or in the zygote. So that here obviously we need to consider a formula like X index, or it is capital I, is equal to number of X chromosomes divided by number of the sets of autosomes, because autosomes carry the genes for the expression of maleness in a Drosophila only. Please try to understand. And here let me refer this table directly so that you will be able to understand it more clearly. And let me tell you firstly what all it contains. This is the first row carrying the titles like number of X chromosomes. Please remember, X chromosomes means that is for the femaleness. And second, this row is for number of sets of autosomes. Why? Because autosomes contain or carry the genes for the expression of maleness in Drosophila. And by considering a ratio between the first and the second column, that is the number of X chromosomes divided by number of sets of autosomes, one set or two sets or three sets, we will see it in the next level. And ratio between X and A, that is X divided by A, will determine finally or in the end as either a male or a female or super male or super female or intersex or tetraploid female or triploid female, something like that. However, I want to make it very clear. Please try to consider, here is an egg, and this egg, you know, it is a haploid cell. In case of Drosophila, the haploid set of chromosomes are only the 4, where 3 of them are autosomes and 1 of them is X, because it is egg as a female haploid. You know that. And here is a sperm. Sperm is a male gamete. Again, it is having 4 chromosomes because it is a male haploid cell. And 3 are autosomes and 1 is Y chromosome. So when this sperm comes and unites with the egg, it forms a zygote. But now consider the genetic constitution or the total number of chromosomes. Egg having 4 chromosomes and sperm having 4 chromosomes, 4 plus 4, totally the 8. So that 4 pairs of chromosome is the 2n number of chromosomes in case of Drosophila. This is known to you people previously. Now you consider when a zygote is formed having 1 X chromosome and how many sets of autosomes? 2 sets of autosomes. So why? Because in sperm there is 1 Y chromosome and 1 set of autosomes, and egg is having another set of autosomes. Autosomes of egg and autosomes of sperm, n plus n, totally 2n, means 3 plus 3, 2 sets of autosomes. The genes present on these 2 sets of autosomes play an important role in the determination of sex of the individual, either into a male or a female. Let us consider the first example. If the zygote containing one X chromosome or allosome and having two sets of autosomes, not two autosomes. Two sets of autosomes means totally six, three autosomes of sperm and three autosomes of egg in a zygote. So two sets of autosomes, so make a ratio, that is 1 divided by 2, that equals to 0.5, and 0.5 genetic constitution determines the maleness in a Drosophila. That becomes a normal male Drosophila. Let me repeat it again. When zygote contains one X allosome or one X chromosome and two sets of autosomes, but here do not try to consider Y, because it is not playing any role in determination of maleness. That's why we are not referring it or we are not mentioning it here. Directly the genes that are all present on the two sets of autosomes will play a role in determining sex of the individual. So 1 divided by 2 sets of autosomes, 1x divided by 2 sets of autosomes, it is equal to 0.5, and this 0.5 figure or genetic constitution refers to maleness in Drosophila. Suppose if this is a case, like when zygote contains two X chromosomes and two sets of autosomes, a ratio between these two, that is 2x divided by two sets of autosomes, that becomes 1.0, and this genetic constitution or genetic strength leads into the expression of the femaleness in Drosophila. This is a normal female. So now we can understand or we can deduce that if the genetic constitution is 0.5 by taking a ratio between X chromosomes and sets of autosomes as 1x divided by 2a, it leads to 0.5, and 0.5 refers to the normal male Drosophila, whereas zygote having 2x allosomes and two sets of autosomes, a ratio between these two, that is 2x divided by two sets of autosomes, that becomes 1.0. Obviously it leads into the formation of normal female Drosophila. And directly coming down to this another one special case, that is the next one. Okay, if the zygote has 3 X chromosomes and 2 sets of autosomes, here you may ask me a question that why there are 3 X chromosomes instead of either 1 or 2. This is because of clearly due to the reason like a chromosomal non-disjunction, where egg might receive, by matter of chance, it may receive 3X chromosome because of chromosomal non-disjunction. Okay, so that if there are 3X chromosomes in zygote and two sets of autosomes, then proportion between these two, that is 3x divided by 2a, 3 divided by 2 leads to the strength like 1.5 genetic strength, and this genetic strength leads into the formation of super female or meta female. Okay, similarly that may be the opposite case where zygote carrying the X chromosome 1 as usual, but the sets of autosomes instead of 2, if there are 3 autosome sets or 3 sets of autosomes, because of chromosome non-disjunction, 1x divided by 3 sets of autosomes, so that leads into 0.33, and this genetic strength leads into the formation of meta male or super male. By means of this, I want to tell you very common understanding is that anywhere the genetic strength above 1.0 leads into the formation of super or meta female, and anywhere genetic strength lesser than 0.5, that leads into the formation of either meta male or super male. Okay, and there may be another special case where the genetic strength is more than 0.5 and less than 1.0, and this is known to be the formation of intersex. There could be the formation of two genetic strengths like 0.75 and 0.67, 0.75 and 0.67. Please try to know how this genetic strength is formed. Look at it. If zygote containing 3x autosomes and 4 sets of autosomes, then obviously 3 by 4, 0.75, it leads into the formation of intersex. This could be neither a female nor a male. This is known to be intersex, in between male and female sexes. So it is called as intersex. Then there could be one more possibility where zygote containing 2x chromosomes and 3 sets of autosomes, so that 2 by 3, that value becomes 0.67. Once again, it is above 0.5, above maleness, and below 1.0, below femaleness. So once again, it becomes intersex. So here the formation of either normal male or normal female, or in between these two as intersexes, I think you people must have understood it. Any questions or queries about it? Because it is little bit difficult. You can ask me questions by forwarding them in a group. But please, here I want, now I remember, please remember, my dear students, by referring this or by listening to this lecture, you might come across the few problems or few questions or difficulties. Please be active in questioning me through the WhatsApp group. So this is all about genic balance theory. Genic balance theory has enabled us to determine sex in case of the Drosophila. Next, let us move on to the next point, or next concept, that is formation of Gynandromorphs. Already I have made it very clear about intersexes. There is no point in knowing it again and again, but let me talk about Gynandromorphs. The name itself implies, or this term, look at it, you split gynandro, split gyno refers to gynosia means female, and andro refers to male, means these are actually the individuals. These are the individuals where the body, part of the body show maleness and remaining part of the individual body shows the femaleness. In a single individual body we find male part as well as the female part. So hence it is called as gynandromorph. Let me make it very clear. Genetically they are said to be like the gynandromorphs are sexual mosaics. Okay, and how this case is understood? This is by means of understanding or considering the reason behind of the formation of gynandromorphs, because gynandromorphs are not the normal individuals. Gynandromorphs are not clearly the male or the female. Gynandromorphs are not the intersexes. It is an individual showing partially the maleness and partially the femaleness, maybe more or less. Okay, and how this is possible to observe? We need to consider a special case where once the fertilization is over, zygote is formed. Once zygote is formed, so as to become the new young one, zygote needs to undergo the further developmental stages like cleavage. Cleavage stages, cleavage is enabled by the undergoing the simple mitosis, that is a duplication in the number of cells from the zygote and without variation in the number of chromosomes. Basically here gynandromorphs arise from the zygote having the X chromosomes, only 2X chromosomes. When they are present, obviously it has to develop into female or female flies only. Again we are referring this gynandromorph case in Drosophila. So absolutely, for the origin of gynandromorphs, zygote must contain 2X chromosome complement. Please remember, when this is a case, then it is normal now, but zygote has to undergo the cleavage. Something goes wrong with the cleavage. Either at the first cleavage or at the second cleavage, the descendant cells of the zygote might lose one X chromosome, so that the next successive zygotic cells, by undergoing the cleavage, would contain only the one X chromosome, by losing one another X chromosome, so that continuously there is a formation of female characters only. So that directly I want to come to the or refer the slide that is here where diagrams are there. It is a diagrammatic representation of the formation of gynandromorphs. So let us consider this is a zygote carrying two X chromosomes. Obviously, this zygote has to develop into a female fly or a female fruit fly or a Drosophila. But what happens during the first cleavage? At the end of first cleavage, there are formation of two daughter cells. But here one daughter cell is known to contain clearly two X chromosomes. But another daughter cell here there is one X and one no X chromosome. That means here one X chromosome, by matter of chance or by some mistake, one X chromosome is lost, so that this zygotic cell has only one X chromosome instead of two X chromosomes. But now we need to consider this diagram. Let us consider this is a zygote or fertilized egg, and it has a line. This is a zygote egg line or axis, division of axis. But the spindle formation in between the this is known as a cleavage spindle or mitotic spindle. If it is present right angle to the axis of the zygote, or this what I am moving the cursor, it represents axis of the zygote. Perpendicular to this there is spindle, mitotic spindle or cleavage spindle, where one is shown with clearly dot that is black and another one with this spindle belonging to the X. So once it divides, look at it, it keeps on. The cell carrying only one X chromosome keep on dividing and forming the cells, and this dark colored cells contain only one X chromosome with normal set of autosomes, and remaining cells, half of the cells, look at it, because of the continuous cleavage, each zygotic cell contains normal XX chromosomes. So XX chromosomes, obviously these cells are responsible for the formation of female characters, and this one absence of one X chromosome, single X chromosome, would lead into the formation of male phenotypic characters, so that half part of the body is male and remaining half part of the body is female. So after continuous cleavage stages, at one point of time this zygote would emerge out into a fly, and here look at it, half part of the Drosophila shows the male dark portion, that is maleness or the male characters, and left half shows the female characters. So this is called as bilateral gynandromorph. Left half showing femaleness and right half shows the maleness, so that it is a bilateral gynandromorph. Then there is one more possibility where zygote carrying XX allosomes undergoing the first cleavage, where the mitotic spindle is not perpendicular to the egg axis, it is parallel to the zygote or egg axis. In this case, here one daughter cell, you can refer this as this, having two X allosomes, one daughter cell, another daughter cell having only one X chromosome and due to loss of another X allosome or chromosome. So that with two X chromosomes, lower half, these all cells are produced out of this cell carrying XX, and this forms of femaleness, and upper half where each zygotic cell due to continuous cleavage with a single chromosome, X chromosome, forms the maleness. And finally, after quite a large number of cleavage stages, what we find, the lower half of the embryo carrying XX containing cells for femaleness, and upper half of zygotic cells containing only X chromosome with absence of another X chromosome, so maleness. So when it hatches out into the young one, this young Drosophila or a fruit fly, showing upper half dark colored as a maleness and lower half of the fruit fly showing the femaleness, so that it is called antero-posterior gynandromorph. Upper half of the means from head region as well as thoracic region showing the male sexual characters, and lower half, that is abdomen, with the wings and the legs and the last terminal end of the segment of the abdomen showing the femaleness. So this is called as antero-posterior. Suppose if it is, I mean, mitotic spindle or this cleavage spindle is oblique or in slant position, neither perpendicular nor parallel to the egg axis. If it is in oblique position, obviously here this region, half of this region of the zygotic cells showing XX chromosomes and femaleness, and the remaining half with only one X chromosome showing the maleness, and finally large number of cells produced where half of them would carry XX chromosomes and remaining half of them would carry only one X chromosome. Please don't get it confused with the set of autosomes. Set of autosomes are quite normal, like in normal number, like that is 3 plus 3. But only case variation here is with XX chromosomes and only one X chromosome. It shows oblique nearly about 3/4 of the fruit fly. After the metamorphosis, what happens, that shows the maleness, and 1/4 of the body shows the femaleness. This is somewhat like almost bilateral, but it is 3/4 of it is bilateral and remaining part is only the femaleness. And there could be one more case where the problem doesn't arise, that is the loss of one X chromosome in zygote cell doesn't arise or occur at first cleavage stage, but it might occur at second cleavage stage. At the end of the second cleavage, you know that there is formation of 4 daughter cells, but at the second cleavage there might be the occurrence of this error where there is loss of one X chromosome, and subsequently because of the continuous cleavage, look at it here, only the 1/4 of the entire embryo showing the cells with the loss of one X chromosome, and 3/4 of the embryo showing the cells containing 2X chromosomes. And after metamorphosis, what happens, the entire this Drosophila or this fly showing the anterior half. If you consider this is anterior and this as a posterior, this anterior half where left half of it showing maleness and left half of it shows femaleness, and obviously this is something different, like anterior part only shows the bilateral gynandromorph and the lower half shows clearly the female gynandromorph. Okay, and this is the way how exactly the gynandromorphs are formed. Let me conclude this concept, like what are gynandromorphs? Gynandromorphs are like actually the genetic mosaics, where this case are the gynandromorphic individuals in case of fruit fly showing part of the body as a female and remaining part of the body as male, or vice versa. And this is all because of the error that occurs or that takes place during the cleavage after the formation of zygote. By undergoing the first cleavage or second cleavage, there might be the loss of one X chromosome, so that here there is formation of two kinds of cells, whereas one set of cells due to continuous cleavage formation of XX chromosomes as femaleness, and another set of cells having only one X chromosome with loss of one X chromosome, obviously that forms the maleness. Hence this is all about the gynandromorphic individuals, and once again we refer Drosophila as an example for it. But this is not a normal phenomenon, and intersexes, I do not want to repeat it again. I have made it very clear while referring the in the previous slide at table. Okay, please refer it. Once again, briefly, I want to cover up only this second point. This second point is telling you that the sex ratio for the intersexes could be either 0.75 or 0.67. Here It is because 0.75 is because of zygote containing 3 X chromosomes and 4 sets of autosomes. Then one more intersex case is due to the 0.67 sexual genetic strength. That is because of the presence of 2 X chromosomes and 3 sets of autosomes. Intersex is formation. However, the gynandromorphs or the intersexes in case of Drosophila are not biologically viable. They cannot live and they are found to be sterile and their generation cannot be continued. Usually their origin occurs during the cleavage stages or during the formation of the zygote. And moving on to the next slide to consider the third concept. I hope you people must be following my lecture. And coming on to the next concept, sex determination rather than chromosomal mechanism, rather than considering the type of chromosomes present in it. After the birth, the individual's sex could be reversed, could be changed. How that is possible? We need to consider the different cases, different examples, because these all wonders, they do occur in the nature or in the environment. That's why we say that nature is the supreme. Even by referring the case of COVID-19 virus, nature is the supreme. Nature has proved that it is the supreme power in regulating, in limiting the population of the living species. That's why nature is a limiting factor itself as a superpower. However, coming to the point or the concept title, that is effect of environmental factors on determining sex of the individual. Okay, and let me take the examples one by one. Here is one example, that is a marine annelidon called as Ophiotrocha. In this Ophiotrocha, what happens? Zygote is formed already, no doubt. But the case is that here, once zygote develops into a male young one, this male young one, if it is known to be a male, it will begin to, after attaining sexual maturity, it will begin to produce the sperms as it is a male individual. But what happens as it grows older, as the time is passed by, as it grows older, as it is an annelidon, number of segments will increase in its body. Increased segments number in the body could influence the individual. Obviously, after some time, the sex of the individual will change into female and it begins to produce the eggs instead of sperms. Once again, I repeat, in case of Ophiotrocha, after attaining sexual maturity, it will begin to produce the sperms as it is a male. But what happens over a period of time, it grows older and number of segments will increase. Increase in the number of segments will influence the worm to reverse its sex. Not reverse, change its sex from male to female. And after that, ovaries will develop and it will begin to produce the eggs and it becomes a female Ophiotrocha instead of male. In the very beginning, it is a male Ophiotrocha. At the end, after becoming older, it becomes a female Ophiotrocha. Then how is that proved? This is proved by doing an experiment like the increased extra segments, when they are amputated, when they are cut off, when they are removed, once again sex is back. That is called, it reverts the sex of the individual. Decrease in the number of segments would change the sex of the Ophiotrocha from female to male, and once again it begins to produce sperms. In this way, sex could be changed and also could be reversed experimentally. This is a case of Ophiotrocha. You can have a view on this. This is a marine annelidon, name is Ophiotrocha. Next, one more different example, interesting. It is a wonder of the nature. Wonders are happening in the environment, in the nature, not environment, in the nature. What happens? Here is one more marine annelidon as a Dinophilus. This Dinophilus has got different influencing factor from the nature. How is that? Size of the eggs. size of the eggs produced by the female individual. If the size of the egg is smaller after fertilization, that smaller sized zygote would develop into male. If size of the egg is bigger after fertilization, that fertilized egg would develop into female. So that smaller sized eggs after fertilization develop into male, and larger sized eggs after fertilization develop into female individuals. This is actually a fact that occurs in case of Dinophyllus, which is known to be a marine annelidant. There is one more example. It is a molluscan. It is a bivalvian shelled animal, Crepidula, name Crepidula. In this genus, what happens? Here, particularly the area where it develops after fertilization, look at it. The individuals develop, Crepidula individual will develop or sexually determined as a male already. But this male Crepidula is taken out and raised separately in isolation. It has a tendency to develop into a female. Why? Because in isolation, when this male Crepidula is raised or reared, what happens? Its male reproductive system it will begin to degenerate. In that place, female genital system will begin to develop. Once the ovaries are formed, then obviously it will make a Crepidula to change its sex as a female. This is all because the way how they are brought up, the way how they are raised. This is experimental evidence. I am telling you, not naturally in the nature. Please remember, or by matter of chance in nature itself, if the male Crepidula are isolated, segregated, and if the nature forces it to develop in isolation, then over a period of time, the male Crepidula will get changed into female Crepidula. That means sex of the Crepidula will change. This is in case of molluscans as well as annelidants. Let us move on to the chordates. There also we happen to observe all these cases, that is change in the sex or determination of sex by the influence of nature or the environmental factor. So this is the image picture of Dinophyllus, which is a marine annelidant, and this is an image picture of shelled animal, that is molluscan, that is Crepidula. Let us move on to the next one. In certain reptiles, temperature plays an important role in changing sex of the individual. Suppose in case of turtle, turtle and tortoises and the crocodiles are the reptiles. These are the first completely terrestrial vertebrates. Let us consider the first case where turtle is an example. Here, if the eggs of the turtle are incubated at a lower temperature of about 23 to 27 degrees centigrade, then they obviously develop into the males. And under high temperature, if the eggs are incubated to the level of 32 degrees centigrade, they develop into the females. Similarly, in case of the crocodile, in case of the alligator, the case is something different. If the eggs are incubated at the lower temperature, here in case of crocodile and alligator develop into the female, and under high temperature, if the eggs are incubated, they develop into the male alligator or male crocodile. This is also observed and experimentally proved. Then the next one, there could be the determination of the sex by the influence of certain hormones in specific or particular species animals. There is one animal, that is a marine worm, that is Bonilia viridis, and here is a diagram. You might have come across this. We refer it for various other reasons. something strange animal in the nature evolved. This Bonellia viridis. This is the individual female Bonellia viridis. Why I am referring it as a female? Because the reason that it is something very strange to observe that in case of Bonellia species, or in case of Bonellia species, viridis species, female is found to be extremely larger in size, like nearly about 10 centimeter, whereas the male Bonellia viridis varieties are very much minute or smaller, just 2 to 3 millimeter in size. Naturally, when the eggs are there to develop in the nature, those eggs of the Bonellia, obviously they tend to develop into a female. Suppose the larvae of the Bonellia, which are undifferentiated neither into a male or into a female, what happens here? If the larvae which are sexually undifferentiated, if they are made to develop in presence of female, I want to say this is a portion, entire portion is called as a proboscis, known to be bifid, means it is forked. So it is on this proboscis area of the female Bonellia, larvae which are sexually undifferentiated are placed. Then they begin to get nourishment from the proboscis of the female Bonellia, and gradually later on they migrate down to the genital system, female genital system, where they live as parasites by gaining nourishment, as well as the effect of certain hormones secreted in the female genital system. That undifferentiated larvae will definitely develop into the male individuals. So several larvae will develop into the male by getting nourishment as well as the hormonal effect in the genital system of the female Bonellia. So this is something different. Sex of the larvae, if it is to develop into a male, it has to grow either on proboscis of the female or genital system of the female Bonellia. Otherwise, all those larvae naturally would develop into female only, no males at all. So this is the case. And still there are few more examples I like to cite out here. Coming to this point, that is in case of birds like fowls, like female birds. What happens? Usually in case of birds, we do not find gonads in pair. We find gonads single because it is aerial adaptation. If it is a male bird, it will be having only one testis, another testis is rudimentary or non-functional. If it is a female bird, obviously it has only one ovary functional and another one ovary as a rudimentary. You all know this fact. If we do some experiment, like if we remove the functional ovary from the bird, what happens? The rudimentary ovary would become functional, would begin to differentiate into testis, and the bird becomes a male. It is something different, and this phenomenon is called as sex reversal. Born as a female bird, experimentally when the functional ovary is removed, rudimentary ovary is it begins to differentiate into testis, and obviously bird becomes male, begin to produce the sperms. This phenomenon is called as sex reversal. Then the next one is known to be free martin. What is it? It is special case observed in case of cattle. If a cow gives birth to two cows, they are usually found to one of them would be the female, and that female is found to be intersex and sterile. Female calf is found to be sterile and intersex. Okay, and here what is the speciality of this free martin? Free martin, morphologically or externally, it shows the female genitalia, but internally that free martin female calf has got a male genital system inside. However, it is not viable However, it is not fertile to continue its generation because it is a case of intersex known to be a free martin. Okay, and a person called F.R. Lill worked on this. Why? What is the reason or cause for the formation of free martin calf? Free martin calf is formed because of the twins when there is formation inside the cow's uterus or uterine horn. What happens? Their fetal membranes get fused or united, and that leads into the formation of one sex as female. One sex as female, and another one is found to be intersex as a free martin. Okay, and this all completes the determination of sex by the influence of hormones, by the special conditions influencing the change in the sex of the individual after the birth. Then also there is the formation of gynandromorphs and intersexes, right? And also we have discussed about the XXXY mechanism of sex determination in man, and also XXXY chromosome method of sex determination in Drosophila by referring Genic balance theory, and these all concepts are very much important from examination point of view. Please listen to this video clipping very patiently. Try to understand. If you get any doubt or confusion or difficulty in understanding the content of any slide or in my lecture, please put me questions and please be active on the what is it, WhatsApp. And thanks a lot for patientful hearing. My dear students, take good care of yourself, and also try to take care of your academics, and I hope this will be understood by you, and this completes the whole topic as sex determination. And thanks a lot and all the best. Please take care of yourself. Thank you.